Hi, welcome. My name is Somen Chakraborty. I am a university professor with 9+ years of experience teaching upper division biochemistry courses. So today we're going to look at glycogen. Specifically, we will look at glycogen, how it is broken down, or it is also called glycogenolysis. Previously, we have seen how glycogen is made. So glycogen synthesis from glucose to G6P to G1P, then UTP was attached to make UDP glucose. Then UDP glucose was used by glycogenine to make the 6 to 8 residue primer, which was then taken up by glycogen synthase to extend the glycogen chain. And whenever there is a branching point, then glycogen branching enzyme would come and add the branches, which are the alpha-1,6 linkages. And then again, glycogen synthase would continue to make the linear alpha-1,4 linkages. And this way, the glycogen synthesis will be completed at some point. Also, just a refresher that the glycogen is a polysaccharide, it's a branch polysaccharide. We have alpha-1,4 and alpha-1,6 linkages of glucose, and glycogen is synthesized when we have excess of glucose, which is typically after a meal in the fed state. And the glycogen primarily is stored in the liver and in the muscle. So approximately for a 70 kg body weight, 100 grams will be in the liver and about 400 grams of glycogen will be in the muscle. So the need for making glycogen is so that when we are starving, which is typically in between meal, let's say after you eat dinner, then you don't eat breakfast until about 8 to 10 hours. So during that time, the body, the cells, and the brain specifically needs glucose. So in that time, the glycogen is broken down into glucose and sent to other tissues from the liver so that the other tissues can sustain continuing production of ATP during that starving period. So glycogen is a storage polysaccharide, and the reason to have glycogen, it has a lot of non-reducing ends. So the non-reducing ends can be quickly accessed by the enzyme which breaks it down to make a lot of glucose, and this glucose can be transported to other tissues and brain to supply the fuel when we're not eating meal. So that's kind of the summary of what has been done so far. And so today we will look at how glycogen is broken down. And to break down glycogen, we need two different types of enzymes. So let's start on that. So glycogen breakdown is also called glycogenolysis. So whenever there's a lysis, that means this is a breakdown. And so we're going to break down glycogen, okay? So glycogen has both the linear and branch points. So this is, for example, I just drew a simple linear part of glycogen. So that's a glycogen. And these are our alpha-1,4 linkages. And so the enzyme that breaks down glycogen is called glycogen phosphorylase, or simply phosphorylase, because that was the first phosphorylase enzyme to be discovered. So the enzyme here will be glycogen phosphorylase, or simply phosphorylase. So as the name suggests, it's a phosphorylase. That means it will use a PI, or just a phosphate group, which will be sort of the nucleophile, and you would cleave glycogen chain to make G1P. So here what will happen is this is your inorganic phosphate, which will attack. So this will attack right here, and we're going to cleave that bond. Okay, so that's what will happen. So let's do that. We'll use pi, and we're going to cleave the bond. So in the process, what we will make is going to be so this is our G1P, or glucose 1 phosphate, plus we will make a shortened chain of glycogen. So that is what glycogen phosphorylase does. It will act as a nucleophile. It will cleave that alpha 1 for linkage, and it will release G1P, and then it will shorten the chain. And so this is then the shortened chain will be attacked again by glycogen phosphorylase until you reach a branching point. Okay, so the glycogen phosphorylase will only cleave the linear parts of the molecule. It will not cleave the branching points. Whenever a branching point is encountered, then glycogen debranching enzyme will come, and it will shift about 3 units of sugar to the non-reducing end, and it will also hydrolyze the remaining glucose molecule, and so the glucose is released. So this is how the glycogen debranching enzyme will work. So we're going to see how it actually works. What I have represented by these circles are each of them represents a glucose molecule, and we're looking at glycogen chain. So this part is our linear part, and this is also our linear part, and then here is a branching point. So glycogen phosphorylase will break the linear parts, and then the debranching enzyme will shift residues into the non-reducing end. So we'll see how that happens. Okay, so glycogen phosphorylase will shorten the chain, and until it reaches about 4 units from the branching point. So okay, so what happened here again is that this is a glycogen, this is a glycogen, this is our linear part, this is our linear part, this is a branching point. So glycogen phosphorylase will continue breaking down the linear parts of glucose, so linear parts of glycogen. So we broke down all the blue colored glucose molecules, and then we reached a branching point, which is here. So at this point, about 4 residues from the branching point, glycogen debranching enzyme will come, and it will shift about 3 residues of glucose to the non-reducing end of the other chain. So it will basically take from there and put them here. That's what it will do. And this is how you see now you have this new chain where all the red molecules are now over here. Okay, so glycogen debranching enzyme, that's the first thing that it will do. The second thing it will do is it will also hydrolyze the remaining glucose molecule. So right now here it is just one glucose molecule that is just freely hanging. So the debranching enzyme will also hydrolyze that bond, the glycosidic alpha 1 6 bond, and release free glucose. This is just free glucose. Okay, and so this is again done by debranching enzyme. All right, so the product of glycogen breakdown are going to be, products will be G1P plus free glucose. Okay, that's what we will make from glycogen breakdown. Now the question is, what happens with what we will do with the glycogen breakdown products, which is G1P. So as we know that there are different tissues have different roles. So for example, in the muscle, so the G1P that we produced, it will serve 2 purposes. One will be in the muscle, and one will be in the liver. So the muscle, because muscle contraction needs a lot of energy, a lot of ATP, the G1P that is made in the muscle will directly enter glycolysis in the muscle by first being converted to G6P. So the G1P in the muscle will be converted to G6P, and this will be a mutase, phosphoglucomutase. Okay, so mutase enzyme will convert G1P to G6P, because we know G6P, we need G6P to make glycolysis. So that's what it will do in the muscle. So it will continue into glycolysis and make ATP, right? Because muscle contraction needs a lot of energy. So whenever G1P is made in muscle by glycogen breakdown, it will go on to make G6P, so that can continue in glycolysis and make ATP. Liver, though, however, liver has a different purpose. Liver wants to make sure that, remember, the conditions under which you're breaking down glycogen is the starving condition. Okay, we are not fed, we are starving. So in that condition, other tissues are lacking glucose. So whatever glycogen is broken down in the liver, the job for liver will be to transport the glucose into other tissues, so that other tissues can survive. We know that liver is a big giver. It will give glucose to other tissues before it uses for its own metabolic purpose, right? So that's what liver will do. It will transport the glucose into other tissues. Okay, so what happens in liver is that the G1P will actually go to G6P, which is still the same phosphoglucomutase, but from G6P it will actually be converted to free glucose, and this is our phosphatase, glucose 6 phosphatase, G6Pase. And then this glucose will be transported to other tissues. For example, brain, okay, muscle, etc. Right? So we see two different purposes. One, the muscle, and on the liver. Muscle uses glucose, which, the glycogen breakdown product, the G1P, to G6P for glycolysis, for its own glycolysis, but liver will export actually G6P as free glucose. So it will export as free glucose to other tissues. So now the question is, what is the difference in the muscle and the liver in terms of the enzymes that are present, that in the muscle we don't make free glucose, but in the liver we do make free glucose. So that's the next thing we will look at. So what happens in the liver? Once glycogen is broken down to G1P, it will go to, so from G1P it will go to G6P. Then different things will happen. So on the outside I have cytosol, and I have drawn this as the lumen side of endoplasmic reticulum. So this is the lumen side of endoplasmic reticulum. What happens is that the G6P will actually enter the lumen side of endoplasmic reticulum. Then G6P will be hydrolyzed by the glucose 6 phosphatase, which is present in the inner membrane side. So this is the inner membrane side of the lumen. So it is a transmembrane protein. It is present on the inside. So it will hydrolyze G6P to free glucose. Then glucose will enter blood plasma. So glucose will come here and it will enter. So this is your blood glucose, and it will enter from the ER into the capillaries by this transmembrane glucose transporter. We know it is GLUT2. In the muscle we have GLUT4, but in the liver we have GLUT2. So the glucose that is made by G6P is glucose 6 phosphatase will enter the blood, and it will be transported to other tissues at blood glucose. So that's what happens here. So the key point to remember is that the muscle does not have the glucose 6 phosphatase. Okay, so the glucose 6 phosphatase is present only in the liver, and because of that the muscle does not hydrolyze G6P to glucose, because there is no need, right? What's the point? Muscle wants to use G6P ultimately for glycolysis. So there is no point to break down G6P to glucose. Rather, just use G6P, then make F6P and continue on to glycolysis. But as I explained earlier, liver glucose is made from G6P so that other tissues can use glucose under conditions when it is starving. So that's how the differential distribution of glycogen breakdown product is controlled between the muscle and the liver. Okay, so makes sense. So now we will see, so we have seen glycogen synthesis and we have seen glycogen breakdown. So the question is, how are these enzymes regulated? Okay, so that's the next thing we will see, is the regulation of glycogen breakdown and glycogen synthesis. So first we look at glycogen breakdown. There are two forms of glycogen phosphorylase, phosphorylase A and phosphorylase B. Phosphorylase A is the active form, phosphorylase B is the inactive form. So we'll see what happens. I've drawn two forms of glycogen phosphorylase. One is the inactive form, which is the glycogen phosphorylase B, and the active form, which is the glycogen phosphorylase A. What you see here is that this enzyme has serine residues on the outside. So that's a serine OH side chain, serine OH side chain. And these OH groups are phosphorylated, okay, by a kinase. And the phosphorylation causes a conformational change in the enzyme, which protects the phosphate groups from outside. So they're, these OH groups are on the outside, but when the phosphorylation happens, there's a conformational change which moved the phosphates to the interior of the protein and protects them from what? Protects them from phosphatase. So we'll look at that in a minute. But glycogen, to make the phosphorylation happen, we need a kinase. Remember, whenever there's a phosphorylation, we need a kinase, and that kinase is going to be glycogen phosphorylase B kinase. Conditions glycogen phosphorylase we want to be active. We want it to be active when we're starving. So which hormone is active under starving conditions? So it's going to be glucagon. So glucagon in the liver, and in the muscle there is another hormone which is called epinephrine. Hormones glucagon and epinephrine will trigger the phosphorylase B kinase activity. So once the phosphorylase B kinase is active, then it will add the PI groups on glycogen phosphorylase, right? So that's what it will do. So these hormones will upregulate the activity of the kinase so that glycogen phosphorylase wants active. Because under starving condition, we want glycogen phosphorylase to be active so that it can break down glycogen and give glucose to tissues and help muscle continue its glycolysis. Okay, so once glycogen is broken down and once the muscle returns to rest, then what will happen is that there will be a conformational change again on the enzyme, and it will expose the phosphate groups to the outside so that the phosphatase, and in this case we'll have a phosphoprotein phosphatase. What that will do is that the phosphatase, as we know, the phosphatases will hydrolyze the phosphate bonds. So phosphatase will hydrolyze these PIs and convert the glycogen phosphorylase A to the inactive form, which is the B form. So now we will see what happens in the liver. In the liver, glycogen phosphorylase is also acting as a glucose sensor. So when there is high glucose accumulation in the liver, the trigger, basically the signal, is that the need for the other tissues have been made. The need for the other tissues have been met. So now in the liver, what will happen? The extra glucose will act as a sensor to stop the activity of glycogen breakdown. That's pretty much what it is. So the extra glucose here, which will enter the liver, it will bind these allosteric sites. So these sites are allosteric sites for glucose binding. Okay, so this site and that site, they are allosteric sites. So glucose will bind and it will again induce a conformational change in the enzyme. It will expose the phosphate groups. Okay, so this is glucose bound in these two pockets. It will induce a conformational change. It will expose the phosphate groups. Then the phosphatase will come and it will hydrolyze. So you make the free OH groups, which now is the inactive form. So this is an extra mechanism by which the liver glycogen phosphorylase activity is shut down, and it makes total sense. Okay, so now we will look at the regulation of glycogen synthase. So glycogen synthase also has two forms. One is the inactive form, which is this, and the other is the active form, which is this. So the inactive form is the phosphorylated form. So there are in this case there are 3 OH groups on the surface of the enzyme which are phosphorylated. So 1, 2, 3, and the phosphorylated form is the inactive form, and then the dephosphorylated form is the active form. What we saw in glycogen phosphorylase is totally different, where the phosphorylated form of the enzyme was active and the dephosphorylated form of the enzyme was inactive. In glycogen synthase, the dephosphorylated form is active and the phosphorylated form is inactive. So, so to make the glycogen synthase active from the inactive form, we need phosphatase. So in this case, this is a phosphatase again. So the phosphatase will hydrolyze basically the phosphates. And we know that under what conditions do we want glycogen synthase to be active? We want it to be active when we are fed. Okay, so when under fed condition we have high glucose, high G6P, and we also have high insulin. So all of these are going to upregulate the activity of the phosphatase. But glucagon, on the other hand, we know glucagon is secreted under starved conditions. We don't want glycogen synthase to be active under starved conditions. So that's why glucagon and epinephrine will deactivate. So it will deactivate the phosphatase, okay, which is what I have shown right here. Okay, now one other thing we will see a little bit is how the glycogen synthase converts from the active to the inactive form. So basically what happens is, as you see, there are phosphoryl groups which are attached. So we need 3 of them. We need 3 ATP, and then we'll make 3 ADP. And the enzyme in this case is called glycogen synthase kinase 3, GSK3, okay, glycogen synthase kinase 3. So the glycogen synthase kinase is what is going to phosphorylate the enzyme to convert from the active to the inactive form. And in this case, what will happen is that the insulin, insulin does not want glycogen synthase to be inactive. So the insulin will downregulate the activity of GSK3, right? Because the insulin is secreted under fed condition. When you are fed, we want glycogen synthase to be active so that the excess glucose can be stored as glycogen. So that is why insulin will prevent the inactivation, okay, and it will trigger the activation as we have seen here.